Thanks so much for setting everything up. Really appreciate it.
I keep doing this.
Yeah, exciting. Yeah, I always think about the move it. Well, I'm glad you agreed to that.
Hello, morning. Can you all hear me? Yep. Okay, welcome and good morning, everyone. For today's plenary, we have two speakers. It's a great pleasure that I introduce my past colleagues at the Natural History Museum, London, Archival Curator Jeff Streicher and Reptile Curator Patrick Campbell. They've both been instrumental for and supportive for researchers around the world who are doing collection-based research. And without further ado, I would like to ask Patrick and Jeff to come and give their talk, interesting plenary talk on the role of museums in 21st century herpetology. Jeff, Patrick.
Yes, good morning, everyone. Thanks for attending. And it's a great pleasure to be here. Myself and Jeff are honored to be at this conference. Thank you, the organizers, for inviting us. I will be presenting the first half, and my colleague Jeff will be doing the second. So we're talking about the role of museum collections in 21st century herpetology. The agenda for this morning's plenary is as follows: the history of museums, the history of the Natural History Museum, ENMH in brackets, and how do museums support herpetology, and then we'll be telling you about a few case studies and some users. So I'll be doing the first two, building a sort of foundation, and then my colleague Jeff will be literally taking you to outer space. You'll see what I mean when he talks about it. So, museums have had a long history going back to the 3rd century BC, when the first known museum was opened in the University of Alexandria in Egypt by Ptolemy Philadelphus, the son of Queen Cleopatra and Mark Antony, or so it is claimed. Over the years, however, the museum's culture has spread to nearly every part of the world. The word museum originally stemming from the Greek word describing a place dedicated to the muses, and therefore suitable for learning, discussion, and study. And certainly Aristotle and Pliny must have had private collections. But it was in the age of the Renaissance where men of wealth mostly kept cabinets of curiosities, pretty much like the one on the right there, which is the earliest illustration of a natural history cabinet. So museums are essentially classifying houses where materials are compared and a difference is noted. Natural history museums depend ultimately on their interrelationships between individuals, populations, species, and their environments. And even at this time, we can see the twofold objective of museums, being one to exhibit objects, and the second as a collection for scholars. And we think the first museum, the Natural History Museum in Europe, was probably that of this chap here on the left, Conrad Gesner, in the 1500s, who was a Swiss naturalist. And essentially, he was a classifier as well, and the museum and his library were his working tools. Nowadays, the word taxonomy, of course, is used for classifying animals and plants, from the Greek word taxis, or arrangement. Hence, also taxidermy means the arrangement of skins. Taxonomy requires institutes where thousands or even millions of specimens are stored for comparison and study. Now in Europe, the early museums were privately owned or were the museums of learned societies. But the first public museum in England was that of John Tredescant from 1587 to 1638. And it was based in South Lambeth, in South London, early in the 17th century, medieval England. And it was so popular that it was dubbed the Tredescant's Ark, as opposed to Noah's Ark. But it was not until the end of the 18th century that the really large museums arose. First, the one in the middle, the Bavarian Museum in London in 1755, and then Bullock's Museum at the Egyptian Hall, both in London. And these were pioneers in the use of museums to educate people. But unfortunately, they were very expensive to run, and Lever and Bullock were forced to sell up. But they had at least shown that museums were popular. But they just had to be, but it was also shown that the large museums must be the responsibility of a tax-paying public. Coming closer home now, the Natural History Museum in South Kensington also owes its origin to a private collection. And that of Sir Hans Sloane, very impressive wearing wig there. He was born in Ireland in the year of the Royal Society, in the year that it came into being, and he would later become the president of that society. He was also president of the Royal College of Physicians, and he came to London to study medicine, continuing his studies in France. Then entering the practice of the famous physician Thomas Sydenham. And 2 years later, he made his celebrated trip to Jamaica before settling down to a highly successful medical career in London. An avid, and one would say eccentric collector, he built up an enormous collection at his home, which was built by Henry VIII down in Chelsea. And the Duchess of Portland once said, "I am but just come from Sir Hans Sloane's, where I have beheld many other things than myself, though none so inconsistent." So, on Sloane's death, on the 11th of January in 1753, he bequeathed his collection to the nation, on condition that Parliament should pay for his executors £20,000, mainly for his daughters. The money was raised by lottery, which was a great deal less than the value of the collection, and the bequest was accepted on those terms by an Act of Parliament in the same year, together with the George II Royal Library, etc., etc. And it was eventually opened to the public at Bloomsbury as a British museum in 1759. And a significant proportion of that collection, as you know, became the Natural History Museum. So when he was in Jamaica collecting, he worked as a doctor on the plantations, and it was rumored that he spent between 50 to 100,000 pounds, which is the equivalent of about 33 times as much in modern purchasing power on that collection. Akan men and women, mainly from the present-day Ghana and the Ivory Coast, helped him to assemble some 800 plant specimens, as well as animals and curiosities. And more and more we are seeing museums engage in conversations where diversity and retrospectively giving credit to those who help people in the field are happening more and more, which is not a bad thing. Composer Handel came to tea, and also the Prince of Wales in 1748, who remarked that it should one day be the property of Britain. This collection comprised some 50,000 books and some 79,575 objects, over half of which were connected in some way to natural history. So the first residency of the British Museum here in Montague House, which was a late 17th century mansion in Great Russell Street, Bloomsbury area or district of London. The original had been burned down in 1685, but it was rebuilt in 1700 by Ralph, the first Duke of Montague, after which it was named. And it was used for about 70 years. So on the 15th of January, 1759, the public first entered the new British Museum here, having a good look at Sloane's curiosities, which comprised something like 80,000 items. It became the Department of Artificial and Natural Productions. Virtually all objects were on display, which is a far cry from what we see now, where only about 2% of our collections are on display. James Epson was Sloane's former curator, and he looked after all the objects. And by 1837, the natural history had 3 branches: botany, zoology, mineralogy, geology. And by 1857, these had also become 4 departments under Richard Owen, who was later to become the director, in all but name, of the present museum in South Kensington. Due to further collections growth, it was soon decided that all natural history collections be removed from Montague House, but there were to be endless delays, as always. And eventually, the present site at South Kensington was bought, and as a result of competition, the new design was given over to Francis Falk, 1823 to 65, who was the architect of the Royal Scottish Museum in Edinburgh. On Falk's early death, however, the task was given over to Alfred Walterhouse, in 1830 to 1905, who decided that an early Romanesque style would do it better in view of the numerous terracotta decorations requested by Owen. You don't want to mess with this guy. The building was completed in 1880, and on Easter Monday of 1881, the first visitors entered the newly built Natural History Museum at South Kensington, a part of London that was already an important part or an important site for exhibitions in the arts and sciences. The collection continued to grow, and the first triples from the Captain Cook voyages between 1768 and 80 filled storerooms. And because of British colonial interests overseas, it continued to grow and become the huge collection it is nowadays. Specimens from his voyage and from the voyages that succeeded them was later added to the considerable amount of Cook and other material donated by Sir Joseph Banks. And it was Banks who pioneered the custom of including a naturalist on board the voyages of discovery. And then a century later came the voyages such as HMS Challenger. So there was an enormous influx of material to the museum, and we also had collections from these gentlemen, Charles Darwin, Rothschild, and Winston, and now Alfred Watts. The growth of empire, the popularity of big game hunting, the increased number of those who turn to natural history as a leisure activity, mostly based on collecting. All of these and many other factors, including the growing prestige of museums, ensured that the staff were kept very busy. And by 1860, the museum was pretty much bursting at its seams. And all of this took place at a time when it was thought that a species could be described on just a few specimens. So where would we put all this stuff? There was a constant pressure over housing these collections correctly. The old Spirit building, it was new back then in 1930, now the old Spirit building, was basically a solid black rectangular structure with thick walls and a sprinkler system to combat fire. But now we have seemed to move slightly away from collection buildings designed purely for their care to buildings mostly pandering to aesthetics, as you will see in a while. There was a dumbwaiter lift system to enable aspirators to be transported in between floors. We had a mezzanine floor, as you can see in the second photo, black and white one next to it. And this was seen as revolutionary back then, because it meant that we didn't have to carry aspirators full of alcohol up and down stairs, and that being a fire risk. But of course, when you look back at it now, it doesn't seem that great. There were plastic tanks in the old tank room there, which again seemed revolutionary at the time. And these ventilation tubes above them were to ensure that curators, when opening those tanks, weren't succumbed by the alcohol vapor hitting their nostrils. Of course, the threat of fire has always been a constant threat and serious concern for us as museum staff and collection staff. We remember the devastating effect that fires had on Luton Town, the Institute of Town, some years ago, 2010, how it destroyed one of the largest collection of snakes in the world, over 85,000. And only 8 years later, again in Brazil, Rio suffered the loss of one of their largest museums collections, which contained Egyptian, Greco-Roman art, and some of the first fossils found in Brazil. So we're always improving our collection spaces, and when our old sprinkler system was condemned as being too old and fit for purpose, and of course in the old buildings we didn't have any clear demarcation between lab spaces, offices, libraries, and our laboratories or collection spaces, we felt it necessary to move to another building, and that is the new building on the left here, the Darwin Centre, in 2001. Fortunately, still on the same site in South Kensington, literally only meters away from the old building, so we were cast into darkness as the new building was being erected for about a year. The temperature of the storms is held at a steady 13 degrees centigrade, which is below the flash point of alcohol. The space is well ventilated to remove alcohol vapor. Sensors in the ductwork detect any buildup of vapor and automatically increase the rate of extraction. Having an engineered low temperature solution to the fire risk avoided the need to install alternative fire suppression systems, such as water sprinklers, which are far from desirable when you have storerooms full of priceless museum specimens with external labels, for example. In the Darwin Centre, all the machinery and ductwork are on the outside of the building. That avoids any risk to specimens for any maintenance work that needs to be carried out. Because of the cold storeroom temperatures, each floor also has a small prep lab at the ends of them for the curators or any visitors to work in a relatively warm environment. The Darwin Centre also consists, in the second picture there, of an atrium, which acts as a buffer zone between the two areas. The first part of the building contains the specimens in alcohol, and the other half, the offices, labs, and library accommodation, which is an improvement from the old building. and also helps us to mitigate against fire. The collections are housed in specially designed metal cabinets, which are fitted with adjustable wooden shelving, achieved from the old building, in preference to a new metal shelving which would not provide adequate shock absorbance. And also the wood absorbs spillages more effectively, and wooden shelves are constructed as a 3-layer laminate to reduce warping over time. And the doors are made of steel. And as you can see in the new tank room, we have steel tanks now, in preference to plastic ones, which tend to leak over the weekend, and then we have to leave our homes and come in and deal with it. So steel tanks are definitely an improvement there. Automatic doors to enable us to get in and out of storerooms quickly, with heavy trolleys heavy laden with specimens. And as a special feature in the new building, we have alcohol on tap, which again is something that we all enjoy as curators. As in not drinking, of course, but topping up jars. So the alcohol is pumped from an underground tank outside the building and is linked through a distilled water distillation system, a dilution system that automatically delivers the distilled or desired 80% concentration, saving a whole lot of time. Focusing now just on the herpetology collection, one of the longest maintained collections in the world, from the late 18th century. And there are approximately 175,000 specimens. And while most of these specimens are preserved in spirit, we also have many skeletal, alizarin cleared in stains, stuffed specimens, mounted specimens, and skins. And the collection includes collections made by Charles Darwin, David Livingstone, Lionel Walter Rothschild, John D. Goodland, Malcolm Smith, Frank Ward, Fernand Matast, Jacqueline Dragio, and many others. And some idea of the importance of our collection is that we hold the types of about 50% of all the recognized snake species in the world. And this is probably true of other groups. It is also the largest reptile type collection in the world, with more than 8,000 specimens. We have the primary types representing 1,968 taxa, the most of any museum in the world collection, and it's represented graphically by this green line on the graph to your right there. The blue line is from the Paris Museum, which holds about half the number of representative type taxa than we do. This is another graph showing you the strength of our collections. So as you would imagine, Africa and Asia feature quite high on this graph due to colonial interests in those areas. Museum holds important historical material from these other regions as well, including many types. But we only have about 7,000 records from the North American and European taxa represented there, as opposed to 20,000 plus records from Africa and Asia. So now I'll quickly run through some of the past herpetological curators and custodians. This is a brief history. Starting with this chap, named Gray, who was the keeper of zoology, who was rumored to have a quite fractious relationship with Robin. He was born in Warsaw, in the UK, and studied medicine, shifted his interests to zoology from botany, I think as a result of making an application to the Linnean Society and being rejected. Can you believe? He volunteered at the age of 15 for the museum, so very young, and joined the zoology department in 1824 to help catalogue the reptile collection with John George Children. Published several other catalogues and was employed for 50 years. He wrote nearly 500 papers. And then we have Albert Gunther, 1857 to 1895, who was ranked the second most productive reptile taxonomist of all time. Graduated in medicine, also in 1858, published a handbook of zoology for students in the same year, and he met Gray and Owen at the British Museum, leading to an offer of work. And then he was put in charge of fishes and reptiles by Gray. He classified 2,000 snake specimens, and again rose to the prominent position of the keeper after Gray's death, and he held that position until 1875. Then we have Arthur O'Shaughnessy from 1864 to 1871. He was an assistant to Albert Gunther as a clerk in the spirit room in the old museum in Montague House, in the basement. And he actually, in 1869, concentrated his specialisms on lizards and became a publisher. He published 12 papers, all on lizards, covering 2 genera and 39 species. But he was also a poet in his spare time and issued books on poetry. But tragically, he lost 2 baby sons and his wife shortly after marriage, and he died just aged 37, succumbing to a lung infection. And then we have the young Belgian, George Albert Boulanger, from 1881 to 1920. He graduated in 1876 with a degree in natural sciences, and he worked at the Royal Belgian Institute of Natural Sciences, Brussels, as an assistant naturalist, studying amphibians, reptiles, and fishes. But he made frequent visits to the museums in Paris and in London. And in 1880, he was invited to work in the Natural History Museum by Dr. Albert Gunther. So sometimes it's who you know. Assigned to the task of cataloguing the amphibians in the collection, he became a naturalized British subject. In 1882, he became the first-class assistant in the Department of Zoology and remained in that position until his retirement in 1920. And there are some of his catalogues which we use regularly, almost on a daily basis. Then we have a woman, Joan Proctor, 1917 to 1923. And she developed a very early interest in reptiles and amphibians, and actually came to the museum to volunteer. So again, a shout out to all our volunteers. She had no academic qualifications and became his assistant, working as a volunteer, and Boulanger took a shine to her, took her under his wings, and she produced her first paper at the age of 19 on pit vipers. And when Boulanger retired, she took charge of reptiles. She did a lot of work on early work on Komodo dragons, her favorite creature. And throughout her short career, she described and classified new species as well as cared for and treated animals. She worked at the zoo as well, the Zoological Society of London, where she was celebrated as being one of the first women of science. I don't think she liked the term too much. She just wanted to be known as a scientist and not focus so much on her being a woman. But she's still celebrated today. There is a bust in the Zoological Society of this woman. And a book that came out in 2018, John Proctor, The Dragon Doctor. So she's still being celebrated now. James Clarence Battersby, 1916 to 1961. Again, a young starter. He started when he was about 15, 16, as Boulanger's assistant. But during the latter end of Boulanger's career, that's when his career took shape at the museum. So for successive staff, such as my predecessors, Colin McCarthy and Barry Hughes, there was quite a tangible link here between them and Boulanger, as here was somebody who actually worked with him, still alive, to talk to them about the experiences with Boulanger in the early 1990s. Hampton Wildwood Park in 1923 to 1957, bringing you into the 20th century now, who became the keeper of zoology at the Natural History Museum from 1947 to 1957. And he authored several books or works on snakes and frogs, even discovered a new species of lizards of the Seychelles, called Lysiid virulence. And then we have another one, Alice G.C. Grandison, or Bunty Grandison, as she was affectionately known. She collected or identified 269 specimens, and also wrote the obituary to Battersby. And the most recent retirees, I'll just run through them quickly. Nick Arnold, 1966 to 2000, was specialized on geckos and inserted lizards, describing 36 species and 4 subspecies. We had Andrew F. Gibson from 1961 to 1991. Garth Underwood, of course, not a permanent member of staff, but almost was as if he was, because of his regularity of visits and how he supported and supervised a lot of students, 1 or 2 of which are in the audience. He worked on snakes mostly and supervised no less than 6 PhDs. And then the 2 most recent retirees, Conor McCarthy and Barry Clark. Coming right up to date now with the current staff, myself, David Gower, Sam Loader, Jeff Streicher, Mark Wilkerson, and various other postdocs and students and volunteers. So at this point, we're going to quickly hand over to my colleague, who will talk to you about how we support herpetology.
Thank you very much, Patrick. So Patrick has done a fantastic job setting up the deep history that most natural history museums have, and is, by using an example of the institution we work at, showed us a little bit more about how museums have operated through time, and told you a little bit more about the types of people that have helped take care of them. For the last part of today's presentation, I'm going to bring us to the more modern era and try and share some examples about how museums continue to this day to support herpetology. And it's kind of an interesting question to ask this morning, because if you've attended the talks over the last few days at this conference, you may have already seen this question answered, as I know many people in the room here use museum collections currently to help their research, are planning to if you're early career, or are very well aware of how museum collections have been used. So what I'm going to try and do is give you a description of the breadth with which museum collections can be used. So hopefully for early career people, you may see some new ways that museum collections could help facilitate your research in the future. And for those more seasoned professionals, maybe you'll see some non-traditional uses compared to your research of how museum collections have been used and are currently being used. Patrick already told you how many specimens a lot of museums contain, and I want to kind of really highlight why museum collections support herpetology. It's because they're just massive repositories of empirical data, actual records of what's happened during the history of our planet, and that ranges across a massive gradient from individual specimens and information that's contained about the lives that they led, all the way up to comparisons across thousands of specimens that can tell us things about how the tree of life developed over millions of years of evolution. So how are these vessels of data, how are they contained? You saw some pictures from Patrick's portion of the presentation of collections, but just as a reminder for specifically how herpetology collections exist in many museums around the world, we can have them preserved in alcohol. We can have them preserved as taxidermy. These are typically how the more historical collections are stored. And in the last several decades, there's been a growing emphasis on genetic resource collections in natural history museums. So you see, here's our liquid nitrogen facility at the Natural History Museum. And so these are from recent collections of tissues from animals around the world. Some of them correspond to voucher specimens elsewhere in the collection, and others are collected as part of disease monitoring or as behavioral studies to look at paternity and things like that. You don't have to just visit natural history museums to access this data. All around the planet, there's been an increasing emphasis on trying to have accessibility be not just for people in a particular country where a museum may be located. And here's an example from our own museum of how we're trying to share a lot of the primary data associated with specimens with the broadest audiences possible. So this is our data portal, and here's an example of some types of reptile and amphibian records that we make available to the public. And this is really kind of a crucial resource because this can answer a lot of research questions, because sometimes just knowing what a specimen looks like is very valuable without taking very precise quantitative measures even. But it also can help with the history of specimens and their provenance and things like that. And it's a great tool for planning research as well. So knowing if particular collections are going to be relevant to whatever your research is. We also can access new types of information from the specimen. So we're not just limited to images and archival information about the specimens. Here's just 3 examples of the types of data that are being increasingly collected from museum specimens. So here we have some examples of advanced imaging. Many of you, we've already seen throughout the conference, use CT scanning as a great way to look at obtain information not only about specimens, but also about the lives they were leading, about the reproductive cycles, like you can see with this Nectophrenoides we have here. Those are little skeletons of baby toadlets inside of it. But also to their diets. We can get diet information this way. And another example with a scanning electron microscope image of a larval Sicilian here, which I think is a Scolicomorphus, but we'll have to confirm with Mark Wilkinson. And increasingly also, we're looking at molecular information from collections. So if you can join us later today for a symposium we're having on museum collections, you'll see some great examples of how museum specimens are being used to sequence DNA. And so we actually have archival DNA, and we can look back into the genetic past of different species. But this is also being done with stable isotope analysis to look at environments and things like that. So these are the types of empirical data that can be extracted from museum collections. What can this all be used for, though? So I want to close by talking a little bit about the spectrum of research topics that natural history collections can serve. So as herpetologists, that means that we're kind of united by our interest in a particular group of animals, but we do very different things as research scientists. It spans a massive range from cultural studies to physics and adhesive properties, as we saw yesterday. But I guess that what I wanted to highlight is that the range of research topics museum collections can be applied to ranges from local scale, from individuals to populations, all the way to global, where you can ask questions about how organisms evolved, how they're experiencing change, and things like that. And the different types of topics that we see that museum collections can serve, they kind of, I think, they benefit from the core purpose of museums, which has been built on the science of taxonomy. So the classification and understanding of relationships amongst organisms. Because museum collections tend to be organized in a way where we have biodiversity categorized, it facilitates research where you can look at a particular group, even if your research is not taxonomy. Because of this library of biodiversity setup that museums have, we can ask loads of research questions. And traditionally, the non-taxonomic questions that museum specimens have been applied to kind of relate into these 3 broad categories, where we're looking at evolution, questions around evolution, questions around ecology, and then maybe a little bit more recently, more thinking about global change and how, as Patrick talked about the history, the deep history of natural history collections, without planning it, we actually have these time capsules of empirical data that span one of these massive important events in our planet's history, which is our entrance into the Anthropocene. So we have loads of specimens that come from pre-industrial revolution that are now ideal for understanding how our impact on the planet may have affected the natural world. So these are kind of very, what I would say, biology-related ways that collections can be used, but collections are not limited to that. They're also used for a range of other types of research question, including those in culture, That's not limited only to history of science, but increasingly natural history collections are being used for art and sociology, so to better understand things about ourselves. I'll get into that in just a second. But also conservation to help us with disease ecology and understanding organismal distributions, and also with materials science, so helping us look at the natural world and maybe benefit from different adaptations that plants and animals and other organisms have evolved. So if you haven't seen applications of this before, I thought maybe I'd just provide a few case studies, some examples, some of which have used the collections in the museum that Patrick and I work in, but others using collections all over the world to give you some case studies for how different researchers use herpetological collections. And I'll start with a classic, which is an evolutionary use of collections. And I want to talk about a research paper that Dr. Ranchu Liu Kamai pursued in around 2012 as part of her PhD. Dr. Kamai is collections manager at Field Museum of Natural History. I think many of you in the room know Chun and know that she is a Sicilian biologist. So she studies these limbless amphibians. And she had been doing loads of field work in Northeast India and collecting lots of Sicilians. She was really interested in these Sicilians because there wasn't a lot of historical information about them. So it was with her collaborators, it was comparisons to museum collections that allowed them for the first time to be able to realize the Sicilians in Northeast India were very distinct. And they were only able to find a handful of historical specimens of these Sicilians. Using a combined approach that involved DNA sequencing and extensive morphological analysis, they were able to use comparisons with museum specimens to describe for the first time a new family of amphibians that we now know is called Cichlidae. So this is a classic way museum collections kind of enhance biodiversity research. What about ecological research? So I'll introduce you to Dr. Kate Thomas, a research scientist who is a visual ecologist and was really interested in the evolution of frog vision. And she wanted to specifically ask questions about whether or not the frog visual system may have evolved in relation to, may have adapted to different types of lifestyles that different frogs lead. Now, when you have a big question like that, you need to look at a lot of different species. So museums were a great stop for her to, within a reasonable time period, look at lots of different species and get lots of representatives across the amphibian tree of life. And she did just that. And using exclusively museum specimens, she and her collaborators were able to demonstrate that anurans, in a quantitative way, demonstrate for the first time that anurans have a correlation between their relative eye size and the different habitats they live in, the different lifestyles they're leading. So the main interesting result was that climbing frogs, scansorial or arboreal frogs, they tend to have relatively higher investment in their eyes. So they have proportionally the largest eyes of all frogs. And those frogs with visual systems that they're maybe not the primary sensory system, like those frogs that never leave the water or those that spend their lives burrowing, they tend to have very low investment in their relative eye size. So using museum collections, Dr. Thomas and her collaborators were able to quantitatively demonstrate this and help us understand a little bit more about how a key sensory system evolved across the amphibian tree. Global change. So this is an interesting case study from Dr. Carla Alujevich's research. And Dr. Alujevich and her collaborators used frozen genetic resources that are held at a museum, and specifically at our museum, at the Natural History Museum. They conducted a study on the southern rock gamma from South Africa, a gamma atra, and specifically were really interested in understanding how the territory of individual male lizards might relate to their success in life, and I'll explain a little bit more. So each of these little maps in here represents a home range for a particular male lizard. These are territorial lizards where they spend time defending their territory and things like that. And Dr. Alujevich and her collaborators were really interested in looking at the thermal quality of these habitats, with the idea that they wanted to understand a little bit more about how temperature changes may influence the behavior and ultimately the reproductive success of male lizards. So in the first year of their study, They went out and they took tail tips, which they wonderfully deposited with us at the Natural History Museum. And then in the years 2 and 3, they sampled all the little baby lizards that they found that could potentially be offspring from the male lizards they sampled the first year. They also got to know the male lizards very well, to know how they were using the habitat, to know how good they were at regulating their space and keeping other males out, how often they were head bobbing and doing a bunch of other things. And they found something really amazing using some simple paternity tests, which is that it turns out those males that have what they call a higher thermal quality had statistically more offspring than those that did not. And another way to put this is those that were better at thermoregulation managed to have more offspring. And this also, there was a significant relationship between those males that possibly because of their more efficient thermoregulating were able to signal a bit more regularly, like with push-ups and head bobbing and things like that. So here's a way that museum collections with the frozen resources have kind of helped highlight a scenario where in a planet where temperatures continue to rise and rise and rise, there may be very real consequences to that if lizards have to spend more time thermoregulating, because we can see, at least in this species, there are serious reproductive implications if you are a good thermoregulator. So those are some examples of how museum collections are used with global change. What about cultural change? So this is quite a fun one, and I wanted to highlight just 2 quick things about how museum collections can really help us clarify the history of science, and then also how they're used more broadly to understand ourselves. This is a great example from our own collection of a discovery based on archives and looking at specimen records that Doctors Aaron Bauer and Colin McCarthy made and published in 2010 of rediscovering Darwin's pet tortoise, which had been rumored for a long time to have survived and had been living out a long life somewhere in Australia. But they were able to demonstrate conclusively that the tortoise actually was quite young and prepared as a specimen, and I guess there was a second one as well that ultimately did not survive as a specimen, and we only have this one representative. But they were able to conclusively say this is what happened to this pet tortoise, and that's quite interesting, just to put a little more context into Darwin's collections and travels. Museum collections also inspire art and help us understand our sociology. And I wanted to highlight a really fascinating project being conducted by Dr. Isabel Davis and an artist named Anna Burrell, where they're essentially collaborating on the natural world and how it's interfaced with human reproduction. And one of the examples they picked was the African clawed frog. So many of you will be familiar with Xenopus and the role that it played as a human pregnancy test for quite a while in the mid-1900s. And I really like this quote from Dr. Davis, where she says that, you know, by kind of looking at where specimens came from or where shipments came from, we actually get a lot more insight than just about the animal, but also about the interlocked nature of politics, trade, transport, medicine, and science. So we learn a lot about our interactions, who was sending things back and forth, who had access to this material, and we can learn a little bit more about how human interactions have played out through the ages. Conservation is a really important thing that museums help serve. And I think the key thing I just wanted to highlight was how they serve as a primary reference for distribution data. Many of you in your research will have used these databases that I have displayed up here. Some of the core observations that tell us where species occur in nature come from vouchered museum specimens that have been properly identified. And so, for example, here, this is just the GBIF records of our UK collections at the Natural History Museum, which we've geo-referenced. So I was also kind of advertising this too. If you can use historical information about specimens in the UK, please let us know, because we've got a much better understanding of where they come from now. And also just highlighting a map of life account from one of my favorite species of frog, where these purple cells, many of those they represent museum specimens where we verified the identity of the animal, and we can say for certain that it was collected in this particular region. So this is really key for planning and understanding where different animals, plants, and other organisms live. Now, finally, Patrick started by saying I was going to take you to space, and I'm going to make good on that promise. Mars. I don't think this is a controversial thing to say. Museum collections inform research on other planets, because yes, there's lots of planetary research that happens at museums, but herpetology can also help, and here's how. This is a project that Sunil Dhillon, who's a PhD student at University of Warwick, has recently started working on that relates to the Curiosity rover and future rovers that are being planned to explore Mars. What does it have to do with amphibians and reptiles? Well, biomimicry. So Sunil is quite interested in looking at how different biological structures may help reduce drag in Martian soils. And so he's looking at quite a few things like ostrich feet and things like that, but he's also looking at crocodile scutes. So he's taking surface scans of crocodiles and alligators in our collection and then modeling whether or not those the scutes on their back reduce drag in Martian soils as they may reduce drag in the aquatic environments these animals inhabit on our own planet. So that's the interplanetary part of the talk done. So to conclude, for the two parts that you've seen today that Patrick and I have presented, we'd just like to say that museums have been at the center of herpetological research for centuries. I hope we've done a good job of convincing you that collections are filled with empirical data that can be applied to a wide range of research, and that also museums serve a really diverse community of herpetologists. And I really want to emphasize that again for all of the early career people in the room, that even if you're doing a type of research where you think, oh, a specimen isn't going to help me, associated information from that specimen or environmental samples that museums hold, they may be very relevant. So please do keep museums in mind as you continue to move forward in your careers. And with that, just want to thank a few people, our family at home at the NHM Herpetology Group, also our main funding sources from DCMS at the Natural History Museum in London, and just wanted to shout out for Synthesis, because I've seen several of you here at this conference that have visited our museum with sponsorship from that program, and it really has helped facilitate lots of collections-based research, and we're grateful for that. And I actually want to call Patrick back up so I don't just absorb all the applause at the end here. And thank you all so very much for coming out this morning.
Thank you very much, Patrick and Jeff. We have time for some questions. Do you have any?
Thanks so much for the very thorough history and the stories of the applications. You mentioned the database about is available for researchers that want to have the base information about species that are collected in the museum. But what about the DNA data? Is that also inserted into the database? So the one that are
like DNA
Yeah.
sequences that have already been generated from collections.
Yeah.
So yeah, that would be wonderful someday if we have that type of integration, and I hope we do. At the moment, because it's a relatively small proportion of the collections, and this may not be true for all museums, but for ours, it's a relatively small proportion of specimens that have had DNA sequence from them. So it's really the way to access those is we refer those that inquire about them to either papers they're published in, or oftentimes people will say, oh, we've deposited under this accession number on a repository like the GenBank or EMBL or something like that, and we can send an accession number that you can download the data. But yeah, hopefully the plan is someday to have it all streamlined so that it's your one-stop shop and it goes back to that, the chain of custody takes it back to that specimen.
Okay, thank you.
Any more questions?
I'm sort of interested in the collaborative nature maybe with other museums. So do you still need to collect from other places in the world where there might be other museums that have those collections? Because of technology, we can access that material information. I'm just thinking of a library system. I mean, I borrow stuff from other libraries around the world. So could you collaborate that way, or do you collaborate that way?
We were just talking about loans this morning. Do you want to take this one?
Sure. Yeah, of course we send loans to other museums where people might be working and not able to travel to the UK. And sometimes that works. What was the other part of your question again?
Well, I'm just kind of curious because, like, I would hope that you wouldn't have to necessarily go collect biodiversity in an area where somebody already has that collection.
Yeah, yeah.
Do you know what I mean?
Yeah.
I mean, you don't have space for it to start with, I would assume.
Yeah, so if I can jump in, the general answer is yes, that museums collaborate quite regularly, and not only through sending specimens that are relevant to research that's happening at other institutions, but also through collaborative projects. Like a lot of the on-site research that happens for researchers and curators based at the museum is in collaboration with other natural history museums, or or smaller cultural museums and things like that. In terms of modern collections, I guess it depends on the particular question. If it is a question you have that is related to organisms that are living now, there may be a justification for making responsible collections. But if that question you have can be indeed answered with historical collections, 100%. That's the beauty of our increasingly integrated museum system around the world, is like GBIF and things like that quickly allow us to say, hey, are there relevant collections? And then, I can either request them from the museum on loan, or I can go hopefully visit if I can find some funding for that.
Thank you very much. And another big round of applause for Patrick and Jeff. I have a few announcements to make. So there is still a chance to bid on the conference banner. Bidding closes Friday lunchtime, and the money goes to the Ukrainian Herpetological Society. So the second announcement is the ARC silent book auction closes lunchtime today. So if you already bid on something, so please collect and pay for it today. And the gala dinner is only for registered attendees, but others may be able to join later around after 9:30 in the bar if you're interested. And the entrance is on the west entrance, and you can see that on your maps which you collected on the registration. So that's one. And regarding the presentations, posters, please come to the reception to collect your posters for the first session, and one of the student volunteers may be able to help you. And the rest of the talks for today and tomorrow, please email it by lunchtime today, if possible, so we can put it in the respective folders and we can continue with the program for the rest of the conference. With that, we can go for a break and have some refreshments. Thank you very much for